Good afternoon. My name is Pinar Bayrak Toydemir and I am a medical director in ARUP Laboratories. And I will talk about exome sequencing as a diagnostic tool. And I use exome sequencing in the clinical laboratory for several years. And in this talk, I will talk about and share my experience about exome sequencing. Before I start, I wanted to give a couple of housekeeping items. These announcements for the CE credits. You can go to bioconferencelive.com for the CE credits. And also for the questions, as they come to mind, please submit your questions to me while I'm speaking so that I can address them at the end of my presentation. In this talk, first I will give a brief background about the method, and then I will show you data analysis using an example cases, and then I will talk about guidelines and recommendations about exome testing from professional organizations. Exome sequencing is a result of the investment made from Human Genome Project. As President Obama states, that was a good investment. Every $1 investment returned $140 to the economy. Of course, this did not happen overnight, but it certainly gained momentum in recent years. DNA structure was discovered in 1953. And 20 years later, Sanger sequencing was developed. And it took another 20 years to sequence one human chromosome. However, it just took a couple of years to sequence an individual's genome after development of next-generation sequencing. Exome sequencing is a powerful tool for gene discovery. Over 200 genes have been discovered in a couple of years, and those were mostly research studies, but now exome sequencing actually is accepted as a powerful tool for diagnostic as well. Part of this is the cost. Next generation sequencing cost is dramatically dropping since 2008. In this graph, white line shows the projected cost in years and the green line shows the actual cost. If you look at the last 5 years, cost of sequencing 1 megabase of DNA dropped from $1,000 to 10 cents. Other than cost, the amount of data produced by this technology is also very attractive. So I wrote exome sequencing to the PubMed as a keyword, and then I got more than 200 papers since 2010. This shows the trend. I wanted to get your attention in this last article published in Human Mutation 2013, and authors discussed that next-generation sequencing will be replacing Sanger sequencing in clinical diagnostics. Cost is a really major factor for next-generation sequencing and really can replace the Sanger sequencing in the future. For example, fibrillin-1 gene is one of the large genes, has 66 exons. So let's compare Sanger sequencing of fibrillin-1 gene by itself and as part of the aortopathy gene panel. This is a Marfan and Marfan-like syndromes gene panels, which includes fibrillin-1 gene and 9 other genes. The cost of these two tests are very similar, so most clinicians prefer to order next-generation sequencing assay instead of fibrillin-1 Sanger sequencing by itself. By exome sequencing, we mean sequencing of all exons of the genome, but we are missing some parts of the genome. Those are some protein coding genes, some exons of some genes, non-genic control elements, some microRNA genes, copy number changes, structural changes or sequencing of mitochondrial DNA as well.
Dr. Fred Stratman, please come to the lobby. Fred Stratman, please come to the lobby.
Exonic regions of the genes are parts of the genome we understand best. Although 1% of the genome are exonic regions, 85% of all known disease-causing mutations are located on exons. So exome sequencing costs 1/6 of the cost of whole genome sequencing. So by exome sequencing, actually we are getting the most useful data with the lowest cost. Although selection of cases has a huge effect on the clinical sensitivity of the test, diagnostic yield is around 20% based on NIH undiagnosed disease program. But we should not forget this is a diagnostic odyssey for the patients. Most of these patients have multiple congenital abnormalities, intellectual disabilities, or unexplained developmental delay or declining, and they have already tested negative for many tests. There are some pre-analytical considerations for the exome sequencing, description of clinical findings, and inheritance modes are the key components of the success. Available affected family members and good family history is also very important. There are some analytical considerations for exome testing as well. So like a capturing efficiency or bioinformatic aspects, which I will cover with examples of bioinformatic aspects a lot in the next of the slides. And also there are some post-analytical considerations for exome. This is really the very hot topic with lots of discussions. For example, is there a really negative exome report? Did the patient consent to learn about everything or just the primary findings? There is a need for education, our patients, clinicians, and also payers. So I will come back to these issues and address some of them using the regulations and guidelines. First, I wanted to talk about a little bit methodology. So this is a really long process. It takes about a month, and there are many steps involved, and automation is really necessary in the website of the laboratory to minimize the errors. The first step is sharing the DNA. So you can use several different techniques to share the DNA. You can use sound waves in this example, and you share the DNA around the 200 base pair fragments. The second step is library preparation. So after DNA is fragmented, the ends are repaired and adapters are ligated. You are ligating two different adapters on ends of the fragments. This will allow to paired end or single end sequencing. The goal at the end is to amplify these fragments for sequencing. Here is the example of the successful ligation. In the first graph, you see this fragmented bases around 150 base pair, and after the adapter binding, you see most of the fragments are around 50 base pair. So this indicates a successful library generation. The next step is enrichment. You can use several different capturing techniques. In this example, you see the RNA-based capturing. So the probes are designed to capture all exons in the genome and also exon-intron junctions. So in this example, we have the biotinylated RNA baits and the DNA. When we hybridize biotinylated RNA baits with the DNA, then we use streptavidin-coated magnetic beads to separate the captured DNA from the leftovers, and then we sequence those targeted regions after. And in this example, you see the flow cell, and this is a cluster generation step. Two different adapters are attached to the surface of the flow cell. You see the colors with the green and blue. We hybridize DNA fragments to the flow cell. The other ends of the fragments also bind to the flow cell to make that bridging step. And by several cycles of the bridge amplification, you see on the left side of the bottom of the figure, and it makes a clusters. The cluster formation actually allows us to read several copies of the same fragment. So one fragment, one cluster, and we get several copies of one read. This cartoon shows how the sequencing occurs in the single fragment. Primer binds to the fragment, and one nucleotide incorporates at a time. And these nucleotides have reversible determinator. After binding, the image is captured, and then the determinator is cleaved. This is a one cycle. So if you have 100 cycles, that represents actually 100 base pair reads. So this is an example of the single base pair end reading. This is capture of one of the cycles at the flow cell. And this is how the flow cell looks like during a cycle. If you flip the flow cell and read from the other direction, this is called paired end reading. Read from other side of the fragment too. This allows to increase the read coverage per cluster, more accurate reading and alignment, detect small and large insertions and deletions, and inversions or other rearrangements better if you use the paired end reading at the end. After all these steps, the fun starts. Data analysis is really challenging. So in this slide, I just wanted to show you the coverage and reads. The blue boxes represents the exons of gene, and gray bars represents the individual reads. So here's the zoom in of the same region on using the IGV Viewer program to see the real reads. Again, this gray bar represents the individual reads. In this example, actually, we see a nucleic base substitution. It's from the T to C change. As you see, some of the reads has the wild type allele with the T, but almost half of the reads represent the C allele. So there is a heterozygote change in this example. Data analysis starts with alignment of the reads back to genome. The use of combination of several programs can help to clean up the data, decreases the false positivities and negativities. However, the most difficult step is the interpretation, since there is no software can do this except a human being. After all steps you're done, and you have the variant calls, and at the end, so you have thousands of the variants at the end to analyze for these patients. The variant filtering out, of course, is definitely the first logical step to start with. But the filtering is really tricky, so you have to really set up frequency cutoff, depend on the disease prevalence, disease penetrance, or inheritance pattern of the disorder as well. So some diseases could have a low penetrance, then you have to set up your frequency cutoff really high, that you should not miss the real mutation in the sample. And based on my experience, the most powerful filtering is actually the family members, and I will show you some examples in the next couple of slides. However, we can filter out based on the inheritance model, location of variant, and variant type as well, which I will show you the examples too. Let's look at this stereo. This is a 11 years old male, and he has a global developmental delay, short stature, feeding problems, hypotonia, hypoplastic genitalia, and loss of dysmorphic features, including facial features as well. So he doesn't have any family history, and he has lots of laboratory work is done, and they're all negative, such as SNP microarray, Fish-4D George predictable lead subtelomeric rearrangement, or regression type B syndrome. He has a normal karyotype and normal EEG and brain MRI. And we received this patient sample with the patient and also his healthy parent as well for exome sequencing. So based on this pedigree, we thought we could analyze the data based autosomal recessive pattern, or de novo autosomal dominant pattern, or X-linked pattern. So we analyzed this data based on all these scenarios, and I'll show you the result. Oh, sorry, I didn't know there was a movie there. Sorry. So okay, here is the example using the autosomal recessive inheritance model. So this patient, if we don't use any filter and anything at all, this proband has around 57,000 variants. So on the autosomal recessive inheritance model, you can use the population frequency around 1%. And so we filter out 1% of the population frequency, and also we kept only the variants on coding or splice site regions, and those are the ones that non-synonymous, stop codon, small frame shift, in-frame changes. So we're really looking for a disease and possible pathogenic mutations, and this patient has 915 variants based on this filtering. Then those are, since this is a recessive model, we have to focus the homozygote of the same homozygote mutation for one gene, or compound heterozygote mutations in a gene as well. So this patient has 53 homozygote mutations and 50 compound heterozygote mutations, that possible disease-causing mutations. Then we use the parent to exclude the variants. We filter out the variants if the parent are homozygous of any of those. So, and then from the 53 homozygote variants, this patient now has 8 homozygote variants. And then we use another filtering, and then we filter out the variants if both mutations in the same parent. So we're really looking for one mutation in one chromosome, the other mutation and the same gene should be on the second other chromosome for this patient to be able to disease causing on the recessive model. So this patient had a 50 compound head, but we did not know the chromosomal phase, and when we included the chromosomal phase using the parent's information, then we had ended up only 15 compound heads possible mutations for this patient. And this is the example for the de novo inheritance autosomal dominant model. On the autosomal dominant model, you may actually change the population frequency filtering and then lower the frequency in 1 in 1,000. So we filter out all the variants more than 1 in 1,000. And then we only look the variants again in like we did before, the recessive, coding in splice site regions, and focus on the non-synonymous, stop codon, small frame shift, or in-frame changes. We ended up having 528 variants for these patients. Since this is a de novo, and then 44 homozygote variants, this patient, that actually not that important because you're not expecting to find de novo homozygote change. And also there's a 484 possible mutations in this sample, and we use at this point parent, and we excluded all the variants in the parent if parents are heterozygote or homozygote for this variant, and we ended up having 88 heterozygote variants that could be possible mutation for this proband. So you can see we can illuminate a lot specifically for the dominant model. An X-linked model is kind of the frequency similar to the recessive. We use 1% population frequency, and this model you only focus the variance on the X chromosome, and then those are only 22 for these patients, and if you filter out anything that father carriers or the mom is homozygote, then you're ending up having only 12 variants that could be possible. Okay, then after all these filtering steps, now we have around 150 maybe variants to go over for this patient. And then in the clinical scenario, you really need to focus on the genes that's been published in the HGMD or OMIM, because those are the genes that are being described well and linked to the disorders that will be useful for the patients to interpret. Otherwise, it will be very difficult if there is no functional studies or phenotypic information of the gene, and it will be very difficult in the clinical world to interpret those results. It will be that something is different than research. So in this patient, actually, patient had ARID1B gene mutation, and this is a stop codon mutation. It's a de novo variant. This is the example of the IGV viewer. You see that's a G to T heterozygote change, and we actually use the Sanger to confirm this mutation as well. And okay, if we found one de novo possible mutation, and then okay, the next question is this stop codon mutation, or is this gene is the disease causing for the patient? So there are two basic questions actually need to be answered. Is this gene is well described, like I said before, in the PubMed, Google, or HGMD, is as any phenotypical link? And then after you figure out that answer, then you have to ask the question, is this mutation is well described as well? So you need to look the pathogenicity scoring, SIFT, polyphen, mutation taster, or conservation. Again, in PubMed and Google's are really useful tools to describe and categorize the mutation pathogenicity based on the available information in the research site. If you go back to our patient, our patient had a stop codon mutation in our ID1B gene. So when we look at the PubMed, we found a Nature Genetics article in 2012, and there were de novo truncated mutations in this gene. This patient actually has a coffin series syndrome. So this was a good evidence for us to see this mutation, this gene actually linked to the clinical phenotype. And when we look at this coffin series syndrome to see how much overlap with our patient's phenotype, coffin series is a global developmental delay, short stature, hypotonia, and some learning difficulties that really overlap our patient's phenotype. So we felt very good about for this patient. And since this is a stop codon mutation, it was a little bit easier to classify. And in fact, that there was an article that described the stop codon mutations in this gene cause a coffin series syndrome. So we ended up reporting in this patient the one pathogenic mutation that is predicted to be causative for the patient's symptoms was detected. So here's one tricky thing in the stop codon mutations. Sometimes when you see the stop codon mutations, you can think that's okay, this is the stop codon mutations got to be disease causing, which actually might not be the case for all the time. So every healthy individuals may have a couple of stop codon mutations, and also some of the genes, even the well described disease causing mutations in that genes it can disease cause. If those genes, the disease mechanism is not a haploinsufficiency, it's a gain of function, then actually stop codon mutations may not cause that disorder as well. So you really need to look for the disease mechanism in similar examples to be able to make that conclusion that the stop codon can disease causing. So it's a little bit tricky. Okay, so that was the first example for Coffin-Siris syndrome. And what about imprinted genes? That's really interesting aspects because you really need to analyze those genes in different set of minds. It's not a recessive or dominant or X-linked inheritance. It's an imprinted gene has their own pattern that you have to really look for it. So a patient may carry the same mutation with one of the healthy parents. This mutation can be disease-causing based on which parent inherited the mutation. So here's one example that I think it's very interesting. We had a case with a 3-year-old male, and this patient had growth delay, proportional microcephaly, and autism spectrum disorder, and some multiple congenital anomalies. And parents described this their son as breaking out the smiles and laughter for no reason. So we received the patient and also parent for exome testing. So when we analyze the data, this time we actually, we always look the imprinted genes. So this time we looked the imprinted gene as well. And we found there is a missense mutation in this patient carries. It's a missense mutation in the UBE3A gene. It's actually one of the imprinted genes and for Angelman syndrome. And this healthy mom also carries this mutation. So, and it's really tricky because on the Angelman, if you inherited your mutation from your mother, you can have the disorder. If you inherited the same mutation from your father, so you may not have the disorder. It's the same mutation if you inherited from your mother, and you'll have the disorder. So that kind of explain the mom can be carrier but not have the disorder. So this is a mutation that has been 300,000 in the population, and we use the mutation prediction programs like a SIFT, polyphenol taster to see the pathogeneity, and it kind of supports the pathogeneity and conserved. And you see on the left upper corner of this slide that it's the G2T change, and we confirm this mutation to G2A change, and we confirm this mutation and by Sanger sequencing as well. So, and then at this step, you really require to do more workup to be able to confirm this is a disease-causing mutation. And in this scenario, the grandparent will be good to test for this specific mutation to see the mother inherited the mutation either from her father or her mother. So that will give us information. If she inherited the mutation from her father, that actually supports the pathogeneity of this mutation. If she inherits that from her mother, this actually tells us that this is not a pathogenic mutation. This is actually just a benign variant. If you look at the Angelman syndrome, actually Angelman syndrome phenotype is very much overlap in our patient's phenotype. Angelman patients may have a laughing and some hand movement as well. So this is just an example of the imprinted genes and analyzing in different perspective. Okay, so when you have the exome, you have to know, especially clinicians need to know this, it doesn't mean that you have result for everything. So there are lots of limitations or the exome testing as well. So human exome is not able to completely analyze, as some genes have not been identified that which may not sequenced. Not all pathogenic mutations occur in the exome, although most of them. So those are really the limitations. Some genes, parts of genes, may not be adequately sequenced to allow for confident analysis. And mutations are located in genes with corresponding pseudogenes, some repetitive or high GC regions, large deletions or duplications, and also mosaic mutations may not detectable some of the exome testing. And if you're not really targeted the mitochondrial DNA, and you may not get the result of the sequencing. And chromosomal phase of identified pathogenic mutations may not be identified, and you have to have the parent table to analyze this way. And mode of inheritance, reduce penetrance, genetic heterogeneity could reduce the clinical sensitivity as well. On the reporting side, there are some limitations as well. So only mutations and variants occurring in non-OM genes or HGMD genes or the genes that have been linked to the phenotype could be actually really interpreted better than other genes. So, and correct reported biological relationships among family members are necessary for proper result interpretation. If this been given wrong to the laboratory, the result may actually misleading as well. So an exome sequencing may not identify the cause of the patient's result, even if this is a genetic condition. So those are really the limitations of the exome sequencing. So from now on, I will switch the talk and I'll talk about a little bit guidelines and regulations. About 6 months ago, there were not really much guidelines or regulations that the laboratory can follow, but these days, as we are really good and very lucky that we have several guidelines that we can follow and do validations and regulations. And right now, the CAP has a checklist that we can follow. And American College of Medical Genetics actually published the clinical laboratory standards for next generation sequencing in this month of the genetics in medicine, so we can follow those guidelines. And CLIA, clinical laboratory improvement amendments, also published there, assuring the quality of NGS in the laboratory practice as well. Okay, this is a really, I wanted to talk about a little bit incidental findings. A couple of months ago, American College of Medical Genetics and Genomics published recommendation for reporting incidental findings. This is really a very new and unique guideline. It's never been reported, published such a guideline before. And then this, in this recommendation, they want laboratory return the results of if the mutations in 57 genes, they listed those genes in one by one, and those genes are associated with the increased risk of 24 serious but treatable diseases, even if the clinicians do not suspect the patients have these disorders. So what are incidental findings? The incidental findings are the findings that variants that found by exome and genome sequencing, which are unrelated to the disease of interest. Although majority of them are benign, however, there are some of them could be really disease-associated mutations. And based on my experience, most of the patients may have between 1 and 5 incidental findings that could be actually related with the disorder, which not related to the patient's phenotype. Although it's called incidental findings, this working group recommended the laboratory actively search for these mutations. So it's not accidentally found in the data analysis. They want the laboratory specifically look these 57 mutations in these 57 genes and see if the patients have pathogenic mutations in these genes and report them out. Those are the mandatory reporting for the known mutations for the disorders such as hereditary cancers, Marfan syndrome, long QT syndrome, Brugada syndrome, and certain cardiomyopathies. So, but they really want you to report only the if in pathogenic, non-pathogenic mutations. They don't want the laboratory report if there anything is uncertain or unknown variant in found on these 57 genes. Okay, here is a discussion, of course, and about this recommendation. So where the patient autonomy comes here, and patients' right not to know their result. So I'm sure there will be lots of discussions and will be continue on several years. But this recommendation, a working group is recommending reporting, even reporting in the children as well. So recommendation for sequencing reporting incidental findings not limited by the age of the person being sequenced. So they believe that the prevention of harmful disease outweighs the ethical concerns. So they want report some children's mutations, even though those are the disorders that adult onset disease, some of the such as some of the cancer disorders. So, and despite some discussions about the incidental findings, exome sequencing has a great potential for diagnosing diseases of unknown etiology, possibly leading to improved treatment and patient care, and quality control measures, data analysis, and reporting of incidental findings will continue to evolve and improve. So from right now, I'll stop and I'll be happy to answer any questions if you write them. And if you want to get the credit, then you need to go to this website to collect the credits. Thank you very much. So I'm looking at if there is any question has been submitted so far, and I don't see any question yet. And I'll be waiting a little bit longer to see if you would like to submit a question. Okay, I think I see one question, maybe not. Okay. Okay, here's one question, and what have been your achievements so far in this field? And so, like I said, it's a diagnostic yield is around 20% based on the NIH studies, and based on our group, clinical sensitivity is also around 20-25%. So we really find pathogenic mutation, clear-cut pathogenic mutation in one case in every 5 cases. So we're able to help 20% of the patient as well. And the second question I see here, what do you consider best solution for genome reanalysis? And I just don't know what does it mean, reanalysis. I guess I'm assuming this is like when you sequence the data, you analyze the data now, and a couple of years later, if there is enough publication, and then if the clinicians want, you reanalyze the data with the existing knowledge, and that's really important. And then I do believe that the laboratory may actually develop some test code for just reanalyzing the existing data in a couple of years later, because I believe there will be lots of more publications and research studies may actually help patients pathogenic mutations that you may not find at the beginning. So I think that's very useful, and laboratories should just maybe create a code for that, and then the clinicians can order for that code just for the reanalyzing. But just keep in your mind the technology is moving so forward, so fast, that actually you may actually resequence and reanalyze instead of just reanalyze as well. Thank you very much. And let me see if there's any other questions. And no, yeah, I don't see any other questions. And yep. Any other questions coming in your mind? You can still email me. Okay, they tell me that there is one more for me, but I really don't see it. Okay, I got it. There is one more question. How can you correlate the results in different genetic mutations with the epigenetic aspects? And it's really difficult question. And I guess you mean with the epigenetic aspects is, and it's just like imprinting, but some other methylations, and it's very difficult to correlate those results and interpret those results if there is not enough. But there are some kits now available to look the methylation status of some genes that will be useful to actually incorporate on exome. It's not in the exome, but you have to really do some methylation treatments and then do the sequencing reactions. So that could be useful for the cancer as well. But it's a very hard topic to do on the clinical side. And any other questions? Okay, it looks like there is no other questions. Thank you very much for listening me. I hope that was a useful presentation. Thank you. Have a good day. Oh, there is one. Okay, sorry. Have you ever worked in neurocriminology and genetics? No, I never worked in the neurocriminology area, and all I work is the inherited genetic disorders, so I don't have any experience. All right, thank you very much, and have a good day.